So I am Abhishek, and I am working with Professor Shobhana Narasimhan at Theoretical Science Unit, JNCASR, Bangalore. The title of my today's presentation is Insights from Density Functional Theory into the Formation and Rotation of an Enantiospecific Assembly of Molecular Raffle Wheels. Yeah. This work has been recently published in Angavante Chemi. I will be happy if you kindly go through our work, and I will be available to discuss anytime. So before going into the details of our work, I would like to introduce you to our collaborator. From the left, he is Dennis, then Peter, then Antula. They all work in the molecular nanoscience lab in TUM Munich of Professor Johannesburg. And then she is Professor Shobhana Narasimhan, my PhD guide, and Saurabh is my colleague from our lab. This work has been mostly funded by TUM Global Incentive Fund. They have also funded our visit there. And I acknowledge the computational resource used from TUE CMS and NSM Paramukti in JNCASR and India government. Now, let me introduce you to molecular rotors. Molecular rotor is a class of molecular motors which is capable of continuous rotation under an energy input. So basically, molecular motors and molecular rotors, they are omnipresent in any kind of living organism. If you look at here, this the rigid framework which is giving the stability to the system is called the host framework, and the rotating unit which is continuously rotating inside the pore is called the guest rotor. So if I introduce you to our system, then you can see this yellow highlighted rigid framework is the host framework. Whereas inside there is the rotating unit. If you focus in some of the gray highlighted area, you will see with time they are rotating. That is the guest unit which is rotating inside the pore. And interestingly, in our case, both the host and the guest molecules, they are of same species. That is BPP-COOH molecule. And the host molecules, which is yellow highlighted, they gets deprotonated and form this kind of rigid Kagome network and generates the pore inside the Kagome network, where the guest molecules remain protonated and occupy the cavity of the host framework and rotate. Is it fine? Okay. So this is the BPP-COOH molecule, which has been deposited on silver 111 surface, and it has been annealed after heating to 373 Kelvin. And when annealed, it has formed this kind of pattern, which I have shown in the previous slide. So the question lies, which isomer of BPP-COOH is found on the surface? What is the energy landscape for rotation of the guest molecules? And what determines this kind of energy landscape? And what if the guest molecules also gets deprotonated, which is a good question. And the final thing is, our experimental collaborators have seen the enantioselectivity of those guest molecules. So the origin of enantiospecificity of guest molecules. So this is our atomistic structure. This is the DFT relaxed atomistic structure of our global minimum structure. So we expect this is the global minima. And this is the experimental STM image, whereas in middle, this is the simulated STM image. And if you look, they agrees quite well, or they matches quite well. Now, if you focus on the head portion of the guest molecules, you will see the head of the guest molecule is round shaped and brighter with compared to its the body portion, which is equally visible in the experimental case. And this head of the guest molecule is having a hydrogen bond with respect to the nearest host molecules. So that has been, we have studied that. I will come to that in the later slides. So that another thing is, if you look at the host molecules, you will see the body and the head of the host molecules are equally bright, and the head of the host molecules are not that round shaped like the guest molecules. So that has been studied by our experimental collaborator, the XPS studies. They have seen that these kind of head groups of the host molecules are chemically modified COO tectons. So that's why they are not that equally round shaped or bright like the guest one. Now the question is, which isomer of BPP-COOH is found on the surface? So there are generally two kinds of isomers of BPP-COOH molecules depending on the nitrogen atom positions in this ring. So this is called syn-syn isomer, and when the nitrogen comes upward, this is called anti-anti isomer. So we have seen that this kind of isomer has been found in some kind of Fe coordinated complexes, which kind of show some spin features or some magnetic features in their cell assembly. Whereas we have seen that this kind of anti-anti isomer is favored in the gas phase structure of the molecule. So in our case, what happens? We have performed the self-assembly of both the isomers on top of silver substrate, but we have found that this syn-syn isomer is higher in energy, whereas the anti-anti isomer forming the self-assembly is lower in energy by 1.84 eV per unit cell. And one more interesting thing we can look, the guest molecules of the anti-anti isomer having a 15-degree angle with compared to the nearest host head group, whereas for the syn-syn isomer, the guest molecules is having an angle of around 32 degree. And with experiment, we have found that our collaborators have found the angle of the guest and host head groups are around 15 degree. So if we compare the energy of the two self-assembly formed by the different kind of isomers, we see that anti-anti isomers is having a lower energy formation for the self-assembly, and also the angle is matching quite well. So from here onwards, we will only talk about anti-anti isomer, which is forming the self-assembly. Now, one more interesting feature in this rotor problem is that guest molecules is rotating inside the pore. So here we have tried to understand how the guest molecule is rotating inside the pore and what determines its rotation inside the pore. If you look, this guest molecule is rotating inside the pore, and during the rotation of the guest molecule, it is forming hydrogen bonds. What is hydrogen bonds here? Now, this alternating yellow and cyan lobes is the hydrogen between the guest and the host molecule, which is forming all that places is highlighted by that alternating lobes. And now we want to understand what determines this kind of rotation. So if we calibrate it with the rotational energy landscape of the guest molecule, we see when the hydrogen bond is missing, it's in the higher energy domain, whereas when the hydrogen bond is slowly increasing and forming the most number of hydrogen bonds, then it's the minimum of the energy curve. So we can comment from here that the guest molecules, as it rotates inside the pore, it forms and breaks the hydrogen bond, and that is the driving force inside the rotation inside the pore. So here I would like to I would like to inform you that this kind of rotation is not unidirectional. It can move clockwise and anti-clockwise in both the direction. So now the next question is how our DFT performed energy barrier matches with the experimental energy barrier. So our DFT performed energy barrier, you can see this is 0.944 electron volt, and from the RNES plot, the experimental energy barrier comes out to be 0.95 plus minus 0.7 electron volt. So the energy barrier for our DFT calculation and the experimental quite matches well. Now the question is, what happens if the guest molecule gets deprotonated? We knew that the host molecules were deprotonated, and we have observed that in STM. But what happens if the guest molecules also, STM and XPS studies. Now, what happens if the guest molecules also gets deprotonated? So we can see, if we compare, the black one is the protonated guest, and this orange one is the deprotonated guest. So deprotonated guest is higher in energy than the protonated guest. So this will be less favored. But one interesting fact is that for the deprotonated guest, the rotational energy barrier is much lower and of the order of 100 of milli eV, whereas for protonated guest it was of the order of 0.94 electron volt. So the energy barrier has drastically reduced in case of deprotonated guest molecules. Now in experiment, they have seen that less than 1% of guest molecules is rotating pretty fast. So we can draw an analogy that this kind of deprotonated guests are those guests which is rotating pretty fast inside the pores. Now the fourth question is, from where the enantiospecificity comes into the game or into the play. So we know that silver 111 surface has a threefold symmetry and it is perfectly achiral, whereas the Kagome network is also perfectly achiral, having a threefold symmetry. Now the question is, the enantioselectivity rise from the twist angle between the surface high symmetry axis and the Kagome network's symmetry axis, high symmetry axis. So we have performed, there are two enantiomers. This is the plus enantiomer depending on the hydrogen atom, and this is the minus enantiomers, and we have performed the rotational energy barrier of both the enantiomers, and we have found that plus enantiomers is forming the global minima, which is at plus 15 degree angle, and which is 41 milli eV lower with compared to the local minima found by the minus enantiomers. From here we draw the conclusion that the pores is occupied by the plus guest molecules. And that also has been reaffirmed in the experiment. So coming to the conclusions, we have made and studied a molecular rotor. DFT estimated energy barrier of 0.94 electron volt for a punctuated pyruate of rotors agrees excellently with the experiment. Global minimum position of the guest molecules inside the pore in the agrees in between experiment and DFT. Features in rotational energy landscape correlates to making and breaking of host guest hydrogen bonds. And constant current STM simulated at global minimum structure matches with the experimental STM images. Symmetry analysis explains chiral induction due to twist angle between substrate and host network. And also our study provides an insight into the area of enantioselective organometallic adsorption and molecular rotor. Thank you.
Okay, so I saw there was a question which says, when the guest atom forms hydrogen bonds and is at lower energy state, then why will it go to the high energy state by itself by breaking the H bonds?
Okay. So yeah, generally it gives a feeling like that when the bonds is formed, it's obviously in a lower energy states, it's the stable states, it will never go back to that. But there is obviously a thermal fluctuations in the systems, and those kind of rotation is seen in the room temperature. Obviously, DFT doesn't calculate any kind of system in room temperature, but yeah, we have tried to explain the observations from DFT, and that observation are obviously experimental observations are in room temperature, whereas when they cool down it to 4 Kelvin, that is very lower temperature, they see that gases are frozen, and then only in that frozen temperature they calculate that those angles, because in rotation mode you can't calculate that angle. So that angle analogy, enantioselectivity, those analyses are a low temperature analysis, whereas the rotation analysis is a room temperature analysis, and explanation of DFT tries to map all the low temperature to room temperature. And I think we have tried to answer that question as per our level.
Okay, Bala, you raised your hand. Can you speak? Are you?
Yes, I can speak. Thank you very much. Very nice work. So I had one question about one movie that you had where you showed the barrier as a function of the angle. The first picture, this picture. There is a movie, yellow color and green color. Very nice. How was this movie made? Is it when you did the nudged elastic band or anything like that?
No. So basically, yeah, there was an option to do NEP calculations, but we haven't done that because it was computationally expensive, we thought. And so what we have done at 15-degree intervals, we have we have relaxed the geometry by fixing the angle. So the angle between host and guest will be like some theta. Theta will vary from 0, 15, 30, 45, 60. And there we have relaxed the system. We have calculated the charge transfer plots. And if you see, these rotations are 15-degree interval rotations.
Okay. Very nice. Thank you. Very good work.
Thank you, Bala. Anybody else here?
So how are the host molecules held together? So through non-covalent interactions only, right?
Sorry, I didn't.
How
The
are
host
the
molecules.
host molecules held together?
Yeah. So
is it non-covalent interactions? He said.
Yeah, I think they are non-covalent because not their distance are not in the covalent bond region. Yes. But you can see also here they are also forming kind of that small hydrogen bonds. These are the weak hydrogen bonds, but that is also formed between the host network.
Plot those.
I plot.
No. These are only the bonds between the guest and the host, I think.
No. The host bonds are also plotted. If you see there, it was there. So first we plotted the guest one, but then we finally changed this to the host bonds are also there. This is the host bonds, but they are not exactly
form the host bonds, which form.
Gas phase. Yeah. No. It's not too accurate. Yeah, it's not the very accurate one, but we have tried. The guest host bonds are the pretty accurate. Okay. The accuracy we have focused there, but host bonds are not.
You could accurate. Do it, but no, they're accurate. You haven't plotted them.
Yeah. So if we plot them more precisely, then the interaction strength will increase a little bit between hosts.
My question is, if there are weak non-covalent interactions, why don't they rotate? Why do only the guest molecules rotate?
So basically, the host network is a stable, rigid network formed by the weak covalent interaction. So
I mean, if they rotate also, they'll come back to the same lattice.
the
so to
host
say.
network doesn't rotate.
Yeah, that's my question. Why doesn't it rotate?
Because there's so the host itself has a threefold symmetry.
No, he's asking if the bonds are of comparative strength, why are those not broken and these are broken?
So I'm maybe I haven't studied in that way, but while I was studying this system, I noticed one thing, that these oxygen molecules have a pretty good interaction with respect to the substrate. So the host molecules oxygen groups is having kind of anchoring to the substrate, whereas the guest molecules, one of the oxygen is bare, another one has the hydrogen which is forming the bond. So maybe that may be one of the reasons, but I am not assured, that I haven't studied extensively that to understand that what's happening. But maybe that may be one of the reasons, because I have seen the host is having anchoring with the.
Okay, we're already 10 minutes behind schedule, so I think we should move on to the next talk.